Okay, so welcome back to this next video in which we are discussing G-protein coupled receptors. Okay, so in previous videos what we've looked at is how ligands can bind to G-protein coupled receptors, cause conformational changes in those G-protein coupled receptors, which then allow the intracellular aspect of the G-protein coupled receptor to interact with heterotrimeric G-proteins that are in the off state, where the alpha subunit has a GDP molecule bound to it and at present is bound with the beta-gamma complex. Okay, so the heterotrimeric G-protein can bind to the intracellular aspect of this activated G-protein coupled receptor, and what that leads to is the release of the guanosine diphosphate from the alpha subunit of the heterotrimeric G-protein. Okay, then what happens is a GTP molecule can come in to that guanine nucleotide binding pocket from the cytoplasm. That causes those conformational changes in the switch 1 and switch 2 regions that remove the ability of the beta subunit to bind to the alpha subunit, and that results in the beta gamma subunit cleaving off from the alpha subunit and also from the G-protein coupled receptor and going off and having signaling roles. Then also what the GTP binding does is it reverses effectively the conformational changes that led to the GDP release when the alpha subunit bounds to the G-protein coupled receptor, okay, and effectively oists the G-protein coupled receptor off. Okay, and therefore the alpha subunit cleaves off from the G-protein coupled receptor and also goes off and has signaling roles. Okay, and we've discussed that then the G-protein coupled receptor that's still in this active state, okay, can then go on and activate more heterotrimeric G-proteins in a amplification process here. So you can have one G-protein coupled receptor activated and it can activate a huge number of heterotrimeric G-proteins. Okay, right, so what we're now going to talk about in this video is a process whereby you can turn G-protein coupled receptors off, which have got their ligand bound and are in the active state. There's a way of turning them off, okay, even though the ligand's still there. I don't just mean taking away the ligand, that would obviously turn G-protein coupled receptors off, but there's a way of turning them off even if they've still got the ligand bound and they're in the active state, and stopping them from interacting with more heterotrimeric G-proteins. In addition, this process actually contributes to desensitization. Okay, so we're going to look at the pathway that underlies how you turn G-protein coupled receptors off and how you desensitize the cell. Okay, so desensitization. So what does desensitization mean? It means making the cell less sensitive to a ligand, basically. Okay, so let's say you stimulate some cell with some ligand. Let's say it's noradrenaline, maybe. It's stimulating the beta-2 adrenergic receptor. Okay, and it produces a certain response within the cell, a certain rise in cyclic AMP within the cell. Okay, desensitization is the process whereby if you stimulate the cell for a long time with noradrenaline, okay, so you deliver a huge amount of stimulation, okay, then if you wait a while and stimulate it again with a brief instance of noradrenaline, the response that will generate in the cell will be less because you previously did the huge stimulation, okay, and this involves basically the removal of G-protein coupled receptors from the cell membrane that results from this pathway that we're about to look at. Okay, so we're about to look at a pathway whereby stimulation of G-protein coupled receptors leads to their removal from the cell membrane, and this is the phenomenon of desensitization. Okay, but this pathway also has a role in actually turning the signaling off prior to removing them from the cell membrane. Okay, right, so let's talk about this pathway then. So basically the pathway involves phosphorylating the intracellular aspects of an activated G-protein coupled receptor. So let's draw a basic picture of a G-protein coupled receptor here. Here's the amino terminus, here's the C terminus. And basically, in this pathway, you're going to phosphorylate the intracellular portions, which are the free intracellular loops and the C-terminal domain of the G-protein coupled receptor that is in the activated state. Okay, so it's important to understand that this pathway is only going to affect G protein coupled receptors that are in the activated state, not ones that are in the inactivated state. So only those that have got a ligand bound and are in the activated state are going to be subjected to this pathway. And basically it's going to involve having serine and threonine residues in these intracellular regions phosphorylated by a class of enzymes which we'll come on to in a moment, and then another protein is going to come and bind to these phosphorylated portions basically, okay, and that firstly is going to stop the G protein coupled receptor from being able to activate any more heterotrimeric G proteins, and also it's going to act as the cue to activate clathrin-mediated endocytosis, basically. It's going to bind to adaptor protein 2, which will then bind to clathrin, and you'll start getting the endocytosis of these receptors, and they're going to be removed from the cell membrane and stored in endosomes. Okay, so let's have a look at this in more detail then. So we'll start off with the class of enzymes that is involved in phosphorylating serine and threonine residues on the intracellular aspect of these G protein coupled receptors. Okay, and these enzymes are known as GRKs. Okay, and GRK stands for G protein coupled, that's the G, so G effectively stands for GPC, the G protein coupled. Then the R stands for receptor, and then the K stands for kinase. So it's just G protein coupled receptor kinase basically. Okay, now in humans you have 7 different G protein coupled receptor kinases. Okay, and these are called GRK1, GRK2, GRK3, GRK4, GRK5, GRK6, and GRK7. Okay, and they are all serine threonine kinases, okay? So they phosphorylate serine and threonine residues within proteins, not tyrosine residues, okay? Just serine and threonine residues. Right, so let's just talk a little bit about where these are expressed. So GRK1 and GRK7, those are really only found in the retina. They're really only found in rod and cone cells of the retina, okay, and they're involved in desensitization in the retina, which is important for being able to adapt to very bright and very dark situations, basically. You need to make the retina more or less excitable by light, basically, in different situations, okay? So GRK1 and GRK7 are involved in the retina. Okay. In addition, GRK4 has a very specific location as well. It's found only really in boys, and it's found in the testes. Okay, so that leads the other 4, GRK2, GRK3, GRK5, and GRK6, to service the rest of the body. Okay, right, so let's just draw a little picture of one of these GRKs. So here is the plasma membrane, here, okay, and basically the G protein coupled receptor kinases are attached to the cytoplasmic face of the plasma membrane like so. So here is our G protein coupled receptor kinase, our GRK here. Now how is it attached to the cytoplasmic face of the plasma membrane? Well, the answer to that depends on which GRK you specifically pick here. Some of the GRKs have lipid moieties attached to them, which then implant into the inner leaflet of the phospholipid bilayer and therefore hold the GRK attached to the underside of the phospholipid bilayer. Okay, so this is the answer for many of them. However, some of them don't have lipid moieties attached, so how do they end up attached to the underside of the phospholipid bilayer? Well, they have special domains which are capable of binding to phospholipids, basically. Okay, so here is this special domain. Okay, and this special domain is what's known as a plextrin homology domain, or for short, a pH domain. Okay, and this is a domain that you find in a huge number of different proteins. Okay, named because it was first found in plextrin. Okay, but it's found in a huge number of different proteins, and it's capable of binding to certain special phospholipids within the phospholipid bilayer, and one of the special phospholipids within the phospholipid bilayer that it's capable of binding to is PIP2, phosphatidylinositol 4,5-bisphosphate, which is found in the plasma membrane. I mean, it's quite a rare one. Okay, it's not the most common phospholipid in the phospholipid bilayer of the plasma membrane, but the point is it's there. Okay, so it is there in some degree, and basically plextrin homology domains are very good at binding to phosphatidylinositol 4,5-bisphosphate, PIP2. Okay, right. So those are the two mechanisms by which GRKs can end up attached to the inner leaflet of the phospholipid bilayer, either by having a lipid moiety attached to them or by binding to special lipids within the phospholipid bilayer. Okay, right. So let's now talk about what these are going to do then. So let's draw the plasma membrane again, and we'll have our activated G protein coupled receptor here. Okay, so we'll say this is R star here, okay, and it is now going to get phosphorylated by one of these G protein coupled receptor kinases. So I'll draw the GRK here. Okay, so this is our GRK. Right, so the GRKs, generally there are absolutely loads of phosphorylation sites on the intracellular aspects of the G protein coupled receptor that the G protein coupled receptor kinases can phosphorylate. So generally what happens is these activated G protein coupled receptors get phosphorylated on serines and threonines all over the place, basically, like so in these intracellular portions. Okay, and this means that they are now a binding target for a new protein, okay, known as an arrestin. Okay, so I'll talk about arrestins in just a moment. So here is our G protein coupled receptor kinase in red here. Okay, so let's say something now about arrestins. Okay, so arrestins are special proteins that are capable of binding to G protein coupled receptors once they have been phosphorylated by the G protein coupled receptor kinases, like so. Okay, now in humans you have 4 arrestins: arrestin 1, arrestin 2, arrestin 3, and arrestin 4. Okay, now just like with GRKs, arrestin 1 and arrestin 4 are actually only found in the retina. Okay, so arrestin 2 and arrestin 3 are the ones that are going to be important elsewhere in the body. Okay, now arrestin 2 and arrestin 3 confusingly have other names, and these are quite confusing. So arrestin 2 is also called beta-arrestin 1, okay, and then arrestin 3 is then called beta-arrestin 2. Okay, so these are the names that you're more likely to see actually used for arrestin 2 and arrestin 3. You're more likely to see them called beta-arrestin 1 and beta-arrestin 2, and unfortunately the numbers don't add up whatsoever. Okay, right, so basically what can happen is once your G protein coupled receptor has been phosphorylated in this way, one of these arrestin proteins can come and bind to the base of it, like so. So here is your arrestin protein. Now the first thing this does is it means that the activated G protein coupled receptor is effectively turned off, okay, because now it's got this massive great arrestin protein bound to its intracellular aspect that is getting in the way, basically. This is no longer able to activate heterotrimeric G proteins anymore, so you've effectively turned off the heterotrimeric G protein signaling. However, in a moment, after we've discussed the desensitization pathway, I will come back to this and say, actually, arrestin is now capable of activating loads of other pathways, and this is actually where one of these paradigm shifts is occurring in the G protein coupled receptor literature. Okay, arrestin is actually capable of activating a huge number of pathways that you would not usually associate with G protein coupled receptors. Again, there's growing interest in this. However, classically, the role of arrestin is to block the G protein coupled receptor from activating any more heterotrimeric G proteins, and it certainly does that. And now, what the arrestin is actually going to do is remove that G protein coupled receptor from the cell membrane and take it to an endosome. Okay, so let's discuss this process in brief. Okay, so the process we're going to discuss is a process called clathrin-mediated endocytosis, which is used all over the place in biology, okay? It's not just used here, it's not a process that's specific for desensitization of G protein coupled receptors, it's used all over the place. Okay, and basically this is a way by which we can endocytose our receptor into an endocytic vesicle, which can then be taken to an endosome. Okay, right, so let me discuss this. So, we're going to draw these pictures a little bit smaller, because if you draw them bigger, they take absolutely ages to draw. So, what's going to basically happen is, onto our arrestin here, a complex known as adapter protein complex 2, which we'll abbreviate down to AP2. So this stands for adapter protein complex 2. Okay, which is a multi-protein complex. It's actually composed of 4 separate proteins, but we will just draw it as a square with the words, well, with the letters AP2 written in. Okay, so adapter protein complex 2 is going to bind to the arrestin, and then on top of the adapter protein complex 2, clathrin, the protein clathrin, is going to bind. Okay, now clathrin proteins can polymerize together. They can join together, and when clathrin proteins join together, the way they join together is to make a sphere, basically. Okay, they don't just join together in some disordered way. They join together to make the outer surface of a sphere. Okay, and this is how we're going to endocytose these G protein coupled receptors. So let me show this. So let's have our G protein coupled receptor now just shown as a blob. So it's been demoted down to being shown just as a square here. So this is the G protein coupled receptor. I might colour this in red just to remind you here. Okay, then we'll have the arrestin bound on top of the bound underneath the G protein coupled receptor rather, here in green. Then after that you'll have adapter protein complex 2 bound underneath there. Okay, and we'll have adapter protein complex 2 in blue here. And then now underneath adapter protein complex 2 you'll then have the clathrin, which I'll show like so. Okay, and we'll colour in clathrin in vivid purple here. Okay, so this is the clathrin. Now, the idea is that what's going to happen is you're going to bring in more G protein coupled receptors that are having the same thing happening to each other, well, happening to them as has just happened to this one. Okay, so here are the G protein coupled receptors, which we'll colour in red here. Okay, like so. And then they will again also have arrestin molecules underneath them, which will then have adapter protein complex 2 underneath that. Okay, again we'll colour it in. So here are the arrestin proteins in green here, and then here are the adapter protein complex 2s here. And then they will also have clathrin molecules attached, and the idea is now these clathrin molecules can start to polymerize together basically. Okay, and when the clathrin molecules polymerize together, as I say, clathrin polymerizes in this bent shape. It doesn't just form a straight line of clathrins on a straight piece of membrane. When it polymerizes together, it makes this curvature and it bends the membrane around it. So as it forms this polymer, the membrane has no choice but to bend in like this. Okay, you can't do this on a straight piece of membrane. It has to bend in like this. Okay, and what's going to happen is this process is going to continue on. You're going to continue extending this. You'll bring in other G protein coupled receptors that have had the same thing happen to them, and you'll gradually extend it up. And what will happen is you'll go on to something that looks more like this, basically. Okay? Here, it's now starting to form a real proper bud here. And let me just give you some nomenclature. This structure that we had here, this is what's known as a clathrin-coated, clathrin-coated pit. Okay, it's a pit in the membrane that's coated by clathrin. Okay, now it's going to really start forming a clathrin-coated bud here. Okay, where you're going to have now loads of G protein coupled receptors. So if I show this, here are the G protein coupled receptors in the membrane here. Okay, and again they'll all have arrestin molecules bound underneath them. And then underneath the arrestin molecules, you'll have AP2, and then underneath the AP2, you'll have the clathrin. Now, I won't show all of the arrestin and the AP2, although they would be there in reality. I'll just show the clathrin to get the important bit across. Okay, so you can view those squares here. You can view that as the G protein coupled receptor with the arrestin with the AP2. Okay, now here, these are the clathrin proteins that polymerize together, and they're starting to form this ball, basically, and they really have pulled the membrane and curved the membrane around themselves to form this bud that is going to bud off in a moment. Now you might think, well, that's really simple, I can see exactly what's going to happen now, it's just going to complete the process round and then this thing will drop into the cytoplasm. But it's slightly more complicated than that. Basically, it turns out that the polymerization of clathrin, although it's brilliant, it does all of this, it doesn't actually have the strength to snip this bit of membrane off there. That requires too much energy, more energy than the clathrin can generate, basically, by polymerizing together. Okay, so clathrin just doesn't have the strength to finish this process on its own. Okay, and it requires another protein known as dynamin. So let me just talk about dynamin. Okay, so dynamin is a dimer, firstly. It assembles into dimers and it's functional as a dimer, okay? And a dynamin dimer is often drawn like so. It's often drawn in this banana shape, like so, with the two dynamin proteins here. So this is one dynamin protein, and here is the other dynamin protein. And when they dimerize, they make this banana shape here. Okay, now, the idea is that this side of the dynamin protein, the concave side of the dynamin dimer rather, okay, this has a positive charge. Okay, there are lots of positively charged residues there, okay, and the idea is that this can attach to curved portions of the phospholipid bilayer, because remember the heads of phospholipids, which are the portions that will be facing into the cytoplasm, they're negatively charged, so this is going to be able to bind to the heads of phospholipids, and with this curvature it's going to be able to bind to curved portions of the membrane. So the idea is that this stalk that you have here, okay, by which the bud is still attached to the plasma membrane, okay, this is the stalk here, okay, exaggerated a little bit here, okay. The idea is that these dynamin dimers can come and bind around the stalk here, like so, and what actually ends up happening is they assemble into helices. Okay, so if I show this, here's another one, here's another one, here's another, and these are all dimers of dynamin, not just a single dynamin protein, and it assembles into a helix that spirals round and round the stalk, like so. Okay, so let's just colour in the dynamin proteins. So we'll have dynamin coloured in here in orange, and these are the dynamin proteins here. Okay, and it forms this alpha helix, well, not an alpha helix, but an helix that will encircle the stalk. Okay, and the idea now is that these dynamin proteins that make up the dimer, they actually all have special domains known as GTPase domains, which are capable of hydrolyzing GTP. Okay, so they can take GTP from the cytoplasm and hydrolyze it. Okay, and the idea is that once you've assembled this helix around the stalk, then what happens is the dynamin dimers in that helix start hydrolyzing GTP, and that hydrolysis of GTP down to GDP and inorganic phosphate releases the energy that the helix needs to constrict, basically. So let me show this. Okay, so if we have our stalk here, like so, originally the dynamin helix goes around, and I'll draw it in dashed lines as it goes behind, and then it comes round again and dashed lines as it goes behind, and then keeps going round like so. Okay, so this is the dynamin helix. What's going to happen is once this is assembled, it can then start hydrolyzing GTP to GDP and inorganic phosphate, and when it does that, the helix effectively constricts, and it's going to turn the stalk into something more like this, basically. Okay, like so. So the helix constricts and it becomes much more steep, basically. It ascends this stalk much more steeply. And the idea is that the dynamin is necessary then for the cleavage of this stalk, well, the cleavage of the bud off from the plasma membrane. It's involved in contracting down the stalk so that the budded vesicle can actually bud off completely. Okay, what you will then end up with is a completely coated vesicle. Okay, so here now is our vesicle in the cytoplasm, and initially it will be utterly coated in clathrin. okay, and also the arrestin and the AP2, okay, and what's then going to happen is the clathrin proteins and also the AP2 proteins are going to come off, okay. So initially this vesicle that we produced here, this is known as a clathrin-coated endocytic vesicle. So I'm just colouring in the clathrin here, which is coating the vesicle here. Okay, so this is what's called a clathrin-coated endocytic vesicle. Okay, and then what's going to happen is the coat is going to come off, basically. The clathrin is going to come off and the AP2 is going to come off. Okay, and then what's remaining there will be our endocytic vesicle here with our G protein coupled receptor in it. Okay, now I'm not totally sure of when the arrestin comes off. Okay, it will presumably come off at some point, but I don't know whether it comes off with the clathrin and the AP2. The AP2 and the clathrin definitely come off in the uncoating process. I'm not sure whether the arrestin comes off. Okay, so here is the G protein coupled receptor in red here. Okay, now we've got our uncoated endocytic vesicle. So here it is. And this endocytic vesicle is now going to go to an endosome, okay? And this is a really important point. People often get confused between the difference between an endocytic vesicle and an endosome. An endocytic vesicle is absolutely tiny. It's one of these little bits of plasma membrane that we have endocytosed off. An endosome is a massive, great intracellular organelle. Okay, so let me show this. So if we've got our cell here, okay, the endosome is a membrane-bound organelle. An endocytic vesicle is a tiny little thing that has come off the plasma membrane. That dot that I've put there, that's supposed to be an endocytic vesicle. Okay, this is now going to shuttle G protein coupled receptors from the cell membrane to this intracellular organelle here, and this an endosome is what's known as an early endosome. So the first endosomes that endocytic vesicles from the plasma membrane deliver their cargo into is called the early endosome. Okay, so what's going to happen is this endocytic vesicle is going to fuse with the early endosome, which I'll draw here, and deliver the G protein coupled receptor into the membrane of the early endosome. So remember, everything is now back to front. The lumen of the early endosome is equivalent to the extracellular fluid, and the cytoplasm is still equivalent to the cytoplasm. Okay, so here is our G protein coupled receptor now delivered to the early endosome. And basically, G protein coupled receptors can sit in the early endosome for long periods of time, basically, and you can store them there until you're ready to put them back on the plasma membrane. Okay, so this is how desensitization occurs. When you stimulate a cell with a ligand, okay, what will happen is the cell will remove its G protein coupled receptors for that ligand from the plasma membrane by this mechanism that we've just looked at, and it will move them all into the early endosome. And that's why gradually the response that you get when you continue to stimulate is going to go down and down and down, because the G protein coupled receptors have now been removed and they're all sitting in the early endosome where they can't be stimulated by your ligand. So basically this is the cell tuning down how much it listens to certain signals, basically. Okay, right. Now, what can then happen is either the G protein coupled receptors can be re-put into the plasma membrane gradually. Okay, so what you can do is you can take little vesicles off the early endosome and re-deliver them back to the plasma membrane. Okay, and that process is then called resensitization. Okay, the resensitizing to the stimulus of the ligand. Okay, or these G protein coupled receptors can be sent on for degradation. Okay, so what can happen is you can bud off an endocytic vesicle from the early endosome, and it then goes to another intracellular organelle, another endosome known as the late endosome. It will go into the late endosome for a while, and then finally it will go off in a little vesicle from the late endosome, and it will go to the final place, which is a structure known as a lysosome, which is a much bigger membrane-bound ball than just a vesicle, and it contains loads of enzymes that will then break down the G protein coupled receptor when it arrives in there. Okay, and this process of having the G protein coupled receptors targeted for destruction is called downregulation. Okay, so the cell will decide what it wants to do with the G protein coupled receptors once they've been removed from the plasma membrane. Does it want to have them destroyed, or does it want to have them resensitized and re-put back on the plasma membrane? Okay, right, so those are the different pathways by which a G protein coupled receptor that has gone to the early endosome can go down. Okay, the final thing that I want to say a little bit on, although we're not going to look at any of this in detail, is that there has been this paradigm shift in G protein coupled receptors. This was the classical view, okay, that G protein coupled receptors activated heterotrimeric G proteins, okay, and then that this arrestin pathway was all about just turning the G protein coupled receptor off, stopping it from being able to interact with any more heterotrimeric G proteins, and then also endocytosing it and taking it to the early endosome so that it's no longer capable of even doing anything anymore. Okay, however, there has been a paradigm shift. What we have realized is that arrestin, once it's bound to the G protein coupled receptor in this way, is actually capable of activating a huge number of different molecules that then start up loads of different signaling pathways. So let me give you some examples of molecules that arrestin has been found to be able to activate. Okay, so some very famous molecules here, SARC, okay, which is a non-receptor tyrosine kinase. It's an enzyme that is effectively a tyrosine kinase that isn't part of a receptor. Okay, MAPK, MAP kinase, okay, part of the RAS-RAF-MEK-ERK pathway. Okay, ERK is the other name for MAP kinase. Okay, so a very, very important enzyme in controlling the proliferation of cells. Okay, NF-kappa B, a very important transcription factor in immunology. Okay, and also a final example is PI3 kinase, which is the enzyme which converts PIP2 in the membrane, wherever it is, to PIP3. Okay, so some very important molecules there that can be activated by beta-arrestin, in fact, more generally any arrestin. Okay, so these molecules are usually associated with very different receptors to G protein coupled receptors. Okay, you don't usually associate, for instance, MAP kinase with G protein coupled receptors or PI3 kinase, or in fact any of these. Okay, but these can be activated by arrestins once they have bound to G protein coupled receptors. So this is one of these paradigm shifts that is occurring. We are realizing how much more G protein coupled receptors do than just activate heterotrimeric G proteins. They in fact signal through these pathways which we do not classically think of as being involved in G protein coupled receptors at all. Okay, we think of them generally as being all about receptor tyrosine kinases. Okay, right. So I'm not going to say any more about that because it's a huge topic in its own right, okay? But I'm going to throw it out there that that is there, okay, and it's to be aware of. And we're going to need this understanding that there is this completely other way that G protein coupled receptors signal in order to then go on to the biased agonism that we're going to have a look at in the next video.